Hello students, today we are going to study the incomplete dominance. Okay, so which is basically an exception of the Mendelian law. Mendelian law. Okay, now what is the incomplete dominance? An incomplete dominance occurs when neither of the two alleles is dominant. Okay, means when the dominant allele and the recessive allele both are not fully expressed. The example of incomplete dominance is the snapdragon flowers. Snapdragon. Okay, which is also known as the 4 o'clock plant. The scientific name of the snapdragon is Mirabilis zalapa. Okay. Now, the flower of this snapdragon is the example of incomplete dominance. So, for the testing of incomplete dominance, here scientists choose the red flower and the white flower plant. Red flower and the white flower in the parent generation. Okay. So these are true plants of the parent generation. Now the red flower was denoted as capital R, and the white flower was denoted as the smaller and smaller. And in the F1 generation, it produces the capital R, small r. Okay, now this capital R, small r showed pink color phenotype. Okay, so here it failed to prove the Mendel monohybrid cross, where the capital R was supposed to be dominant and the small r supposed to be recessive. Okay, and the capital R, small r was supposed to be red. Okay, but this is not the case here. Here the capital R, small r represents the pink color phenotype. Now after that, there was a selfing of the F1 generation. Okay, so the selfing of F1 generation means the capital R, small r cross with capital R, small r. So now the Punnett square is here. The capital R, small r, then capital R, small r again. Okay, and here the first one is capital R, then the second one is capital R, small r. This one is also capital R, small r, and the last one is small r, small r. Okay, so here the genotypic ratio is 1:2:1, where 1 is the capital R, 2 is the capital R, small r, and last one is the small r. Okay, but the phenotypic ratio is also 1:2:1. Okay, 1 is for red, 2 is for pink, and the last one is for white. So in the case of incomplete dominance, we can see that 2 alleles of the true breeding, they failed to express their complete character in the heterozygous condition, and they produces or displayed a completely new intermediate expression. Okay, so this is known as the case of incomplete dominance. Okay. Our next topic of discussion is the codominance. In the codominance, alleles lack dominant recessive relationship and both expresses themselves. Okay, so here none of the allele is dominant and none of the allele is recessive. Okay, so both is equally expressed. Okay, now the example of the codominance is the ABO blood grouping of human. Okay, and the gene I is responsible for the ABO blood grouping, and the I is represented by the 3 alleles. And the 3 alleles are IA, then the IB, and the small I. Okay, so these are 3 alleles which represents the ABO blood group. Now we will see the possible combinations. Okay, so this is the parent 1, this is parent 2, and here the offspring and the blood group of the offspring. Okay, so we saw the alleles are IA, then the IB, and the other one is the small I. Okay, so here the possible combinations may are IA means one parent is IA and the other parent is also IA. Then another one is IA IB. Okay, and then again the IA with the small I, then the IB with the IA, then again IB with the IB, and lastly the IB with the small I. And another combination is when 2 parents are recessive, means small I. And from this result, the IA and IA in the offspring will show the phenotype of IA. which represents the A blood group. Okay. Then the IAIB phenotype. Okay, IAIB phenotype will represent the AB blood group. Then again the IA and small I, this phenotype will represent the A blood group, because small I will represent null. Then another one is IBIA. This will also represent AB blood group, because both the allele is present in this condition. Okay, then the other one is IBIB. This will represent the B blood group. Then IB small I. This will also represent B blood group. And lastly, the small I, and this will represent the O blood group. Okay, so here 4 type of blood group can be generated. One is the A, then AB 2, B is 3, and O is 4. So here A, IA, IB, and I, none of them is dominating the other. Okay, so equally all are equally expressed. Okay, so this is all about the codominance. Now we will discuss the dihybrid cross. Okay, so previously we have discussed the monohybrid cross, which means the cross of one pair of trait or characteristics. One pair of trait. Okay, and here the dihybrid means the 2 pair of trait. Okay. Now, the Mendel's dihybrid cross is related with the law of independent assortment, which stated that when 2 pair of traits are combined in a hybrid, the segregation of one pair character is independent of the other pair of characters. So for the test of this dihybrid cross, Mendel selected 2 types of characters. Number 1 is the sheet shape, and number 2 is the seed color. Okay, and the seed shape were round, and other one is wrinkled, and the seed color was yellow and the green. Okay, and he represents the round as the capital R, wrinkled as the small r, because capital R is dominant and wrinkled character or small r is the recessive character. And here the yellow as Y, capital Y, and the green as small y. Okay, because capital Y means the yellow is dominant. Okay. So here the parent generation means the true plants were the combination of round yellow means the means the capital R, capital R, capital Y, and also the wrinkled green, wrinkled green. Okay, this was represents as the capital R, sorry, smaller, smaller, small y, smaller, small y. So capital R, capital R, capital Y was pure dominant, and smaller, small y is pure recessive. Okay. Now, from this, in the F1 generation, these gametes produced the capital R, small r, capital Y, small y. Okay. And this structure produced 4 gametes in the time of selfing. Okay. Now, the number 4 was the probable combination of this capital R, small r and capital Y, small y. Okay. So, we know that in monohybrid cross, there were 2 gametes, and in the dihybrid cross, we can see the production of 4 gametes. So this is related with the formula, which is 2 to the power n, and here n represents the character number. Okay, the trait number or character number. So in monohybrid cross, it is 2 to the power 1, means 2 gametes. Then in the dihybrid cross, it is 2 to the power 2, means 4. So also we can calculate, then in trihybrid cross, it will be 2 to the power 3, means 8 gametes. Okay, so this is the formula behind the number of gamete calculation. Okay, now let's come back to the dihybrid cross. Okay. Now, in the F1 generation, the characters will produce the capital R, small r, capital Y, small y. Okay, and this capital R, small r, capital Y, small y will produce 4 gametes, and those will be capital R, capital Y, then capital R, small y, then small r, capital Y, and lastly the small r, small y. Okay, now if we do the Punnett square of these 4 gametes, this is 1, 2, 3, 4. Okay, now if we do the Punnett square of this 4 gamete, it will look like this. So this is capital R, capital Y. Okay, so this will be, the next will be capital R, small y, then small r, capital Y, and here the small r, small y. Similarly, in this side, here the capital R, capital Y, then the capital R, small y, then here the small r, capital Y, and lastly the small r, small y. Okay, and here the combinations will produce capital R, capital R, capital Y, then capital R, capital R, capital Y, small y. Here capital R, small r, capital Y, then capital R, small r, capital Y, small y. Okay, and here similarly capital R, capital Y, small y, then capital R, small r, capital Y, capital Y. Then again here capital R, small r, capital Y, small y. Okay. And here the combination will be capital R, small r, capital Y, small y, and here it will be capital R, small r, capital Y, small y. Okay. And in this place, the combination will be capital R, small y, small y. Here it will be capital R, small y, small y. Here and here capital R, small y, small y. Okay. Then in this place, the combination is capital small r, capital Y, capital Y. Here small r, capital Y, small y. Here same small r, capital Y, small y. Okay. And lastly, small r, small y, small y. Okay. So here the red ones representing the round yellow. Okay. The blue one is representing the round green. Okay. Then the brown one is representing the wrinkled yellow, and lastly the black one represents the pure recessive, means the wrinkled green. Okay. So we can see that the red one is present here. This is 1, 2, 3, 4, 5, 6, 7, 8, and 9 in 9 number. Okay. Then the blue one is present here 1, 2, 3 in the 3 number. Then the brown one is similarly 1, 2, 3 in the 3 number, and the black one is the single one, means the pure recessive is in number 1. So here the phenotypic ratio will be 9:3:3:1. So this is the phenotypic ratio of Mendel's dihybrid cross. Now from the Punnett square we can see different genotypic ratio, and those are capital R, capital Y is present in number 1. Capital R, capital Y in 2. Capital R, capital Y, small y in number 2. Capital R, capital Y, small y in 4 number. Small r, capital Y in 1. Small r, capital Y, small y in 2 number. Capital R, small y in 1 number. Capital R, small y in number 2. Lastly, the small y in number 1. Okay. So these are the genotypic ratio of the Mendel's dihybrid cross. Mendel's dihybrid cross. So the dihybrid cross represents the genotypic ratio of 1:2:4:1:2:1:2:1. Okay. So this is all about the part 2 of principle of inheritance and variation. We will see the exceptions of Mendel law and the chromosomal theory in our next class. Thanks for watching.
To learn difficult concept of physics, chemistry and biology within few minutes to crack NEET and JEE with fun, like, share and subscribe our new YouTube channel ShantU. You will get conceptual videos, highly effective notes, short tricks, mock tests, and much more. Hope we shall meet there.